Welcome all. Today we are going to discuss quaternary level of protein organization, which is the highest level of protein organization and is present in many proteins, especially those with higher molecular weight and composed of multiple polypeptide chains. Each of these polypeptide chain is called a subunit. These subunits could be identical or they could be different from each other. If these are identical subunits, these are called oligomers. Based on the number of oligomers, based on the number of subunits, these proteins are named as dimers, trimers, and tetramers. Quaternary structure primarily describe stoichiometry, that is the number as well as their relative position of these subunits in mature protein. Hemagglutinin is a trimer protein that consists of three identical subunits held together by non-covalent interactions. This long multimeric molecule has three identical subunits. Each of these three identical subunits contains two polypeptide chains called HA1 and HA2. The tertiary structure of these polypeptide chain or these HA subunits constitute folding and helices strand into a compact structure which is roughly 13.5 nanometer long and could be divided into two distinct domain, one membrane distal domain which has a globular conformation, whereas the membrane proximal domain that has fibrous stem-like conformation. The quaternary structure of HA is stabilized by lateral interaction between the long helices, that is these cylinders, of the three fibrous domain protein. All the HA subunits has been shown here by three different colors, that is the yellow, blue, and green. And as a result, this hemagglutinin protein, has a triple stranded coil stalk. The multimeric nature of many proteins is crucial for the mechanism which they are performed because they are required to regulate their function. The primary reason for these multimeric assemblies are because synthesis of separate subunits of the proteins are more efficient than to synthesize a substantial long polypeptide chain. Secondly, smaller subunits, smaller worn out or damaged components of the protein subunits are could be effectively replaced compared to the long full length protein polypeptide chains. Thirdly, the interaction of these polypeptide chains help regulate the protein biological function. Not only the multimeric proteins have this level of organization, sometimes different proteins could also associate among themselves to give rise to various functions, and these are called molecular machines. This is the highest level of protein structure, and it's the association of proteins into macromolecular assemblies. The examples are numerous. For example, the nuclear core complex, which consists of roughly 50 to 100 nucleoprotein and is the fundamental to nuclear import export. The capsid protein encases the viral genome. Proteasome machinery is important for to maintain the protein turnover into the cell and to degrade different protein. Not only this, molecular machines are playing very important role. For example, repliosome machinery, which is one machine which contains helicase, DNA binding proteins, primase, DNA polymerase, and the sole function of this machinery is to duplicate the genome. So as soon as the DNA enter into this blue machine, which is the helicase, it spins the DNA like a super jet engine and convert it into two single strands. One of the strand is continuously replicated while other is not continuously replicated because it has to replicate in reverse. That is why it makes loops and turns, and as a result, the overall patch of the DNA is duplicated. Similarly, transcription machinery is there to perform one function, that is the transcription, and it contains helicase, transcription factors, DNA binding proteins, RNA polymerase II. Transcription factors and DNA binding proteins precisely place helicase onto the gene promoter, the place from where the transcription has to start, and then helicase perform its usual function, that is unwinding of the DNA, and make two single stranded copies of the DNA, and then RNA polymerase to move along the single strand to give rise to mRNA, which later on is going to be translated into mature protein. As discussed in previous lectures, non-covalent interactions such as hydrophobic effects, electrostatic interactions, and hydrogen bonds play a very vital role in stabilizing the tertiary structure. Same forces are also important to stabilize the quaternary structure. As for the tertiary level of protein organization, hydrophobic effect are major forces in the interior of the protein. Similarly, when different interfaces of the subunit protein join together, they give rise to these hydrophobic pockets, and these hydrophobic pockets are again there to stabilize the quaternary level of protein structure. Although not very common, but covalent interactions are also present in at quaternary level of protein organization. The examples of these covalent interactions are the disulfide bridges, which are very common in immunoglobulin proteins, where different polypeptide chains containing cysteine residues join together to give rise these disulfide bridges, and these disulfide bridges in turn stabilize this overall immunoglobulin protein and conform and impart it the activity they are performing in the cells. Same is the case with the uh, covalent cross-linking. These covalent cross-linkings happen among various amino acid residues in the polypeptide chain. The common example of these covalent cross-linking is the desmosine, which is the result of cross-linking of 4 different lysine residues. They give rise to this unique desmosine residue and lysino-norleucine residue, and this overall process goes through the oxidation of lysine. Desmosine residue are common in elastin protein, and these elastin protein is uh, frequently present in various connective tissues. It's a fibrous insoluble protein which is not a glycoprotein and present in collagen in connective tissues. The connective tissues which are rich in elastin protein, they have a characteristic yellow coloration. Elastin is a highly branched structure, and this highly branched structure is responsible for physiological elasticity of this protein. These elastin fibers are capable of stretching in 2 dimensions and are found in tissues that are under continuous pressure and tension, as well as in continuous pressure of physiological deformation. The example of such tissues are aorta and other vascular connective tissues, ligaments, and lungs. Normally, elastin fiber is relatively rich in non-polar amino acids like alanine, valine, and proline. However, some polypeptide regions are also have repeats which are rich in lysine residues, and these repeats which are rich in lysine residue provide an excellent place for this cross-linking. The examples of these repeats are lysine, alanine, 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 lysine, and lysine, alanine, alanine, lysine. Mature elastin contains these cross-linked glycine in the form of desmosine and isodesmosine. This cross-linking takes place in the outer extracellular environment of the cell. So the elastin protein is produced within the cell. So as soon as this elastin is produced in the extracellular matrix, it goes through a key oxidative deamination step, and as a result of this oxidative deamination, the lysine residue is converted into a lysyl residue, which in turn is converted into an L-lysine residue by the action of enzyme lysyl oxidase, and this overall process converts lysine into a reactive aldehyde species, which is called L-lysine species. So the combination of L-lysine and lysine is imperative for the formation of this desmosine, and this in fact interact to give rise this cross-linking. Normally, at least two residue, one of which should be L-lysine and one normal lysine residue is required to give rise to desmosine, but four lysine residues, three of which are L-lysine aldehyde residues and one normal lysine residue, joined together to give rise this pyridinium ring, and this pyridinium ring is the result of cross-linking of these four amino acids. So end result is the formation of this desmosine residue, which connects two to four different polypeptide chains. So this desmosine residue could also be formed uh, among different lysine residues of the same polypeptide, but it could join together two to four different polypeptide chain, and as a result, these polypeptide chains are interbranched, and this gives the elasticity to the protein. In routine, 95% residue in elastin are normal, but only 2 to 3 desmosine residue are responsible for the elastic nature of this protein. As discussed earlier, quaternary level of protein organization depends on the association or interaction among various subunits of a multimeric protein. Quite often, these interactions among various subunits are affected by binding of ligands, which results in slight change in protein conformation, which is in turn could affect the activity of the protein. This overall process is called allosteric regulation, and it is a very key process in regulating various biochemical processes. Broadly speaking, allostery refers to any change in tertiary or quaternary structure of the protein or both as induced by ligand molecule, which could be an activator, inhibitor, or substrate, or all the three. These ligand-induced conformational change in protein are called allosteric transitions, and these ligands that trigger these changes are called effectors or modulator molecule. These allosteric effects could be positive or negative depending on whether protein binding increases or decreases protein's affinity to further bind with other ligand molecules. One of the best understood example of allostatic regulation is reversible oxygen binding with hemoglobin molecule. As we all know, what is a hemoglobin protein? This is a oxygen carrying protein in the body, and this protein carry oxygen from lungs to the various body parts. There is another oxygen carrier molecule in the body which is called myoglobin, and it works more or less the same way as hemoglobin because it also has a porphyrin ring which can bind to oxygen. However, it is not a very efficient oxygen carrier because it do not lose oxygen quite easily. That is why hemoglobin is one of the major oxygen carrier molecule in the body. It's a tetrameric protein made up of two alpha chains and two beta chains as it is shown here, and each alpha chain is roughly made up of 140 amino acids and beta chain 153 amino acids. So you can normally say it's an alpha 2 beta 2, that means two alpha and two beta chains, and each of these chain is able to bind to a heme group. This is an heme group. I mean, you can we have already seen a heme is an porphyrin ring made up of different pyrin rings, and जिसके अंदर एक iron molecule center में मौजूद होता है. This is a heme group. Each of these oligomeric subunit is bound with the heme group, and that means each of these could be bound to oxygen. Hemoglobin has 4 oxygen binding sites, so that means it can go through quite a bit of allostatic change. There are other types of hemoglobin as well. Let me just give you an information. For example, approximately 2% of the human hemoglobin is not in fact alpha 2 beta 2, rather it is an HbA2 hemoglobin. And why? Because it contains an other subunit which is called a delta subunit. This is बीटा चेन जो कि आपको नीचे नजर आ रही है, इनकी जगह पर डेल्टा सब यूनिट होगा. दिस इज अनदर टाइप ऑफ hemoglobin which do present in 2% in human body. There are other hemoglobin molecules which are present before birth and soon after birth. So these are two different types of hemoglobin. For example, one is the epsilon chain containing hemoglobin which is present in embryonic life, and there is another one which is gamma chain unit which resembles somehow to the beta chain, but it is different than beta chain. And you can name them A2 epsilon 2 and A2 gamma 2 hemoglobin, and these are also present in the before birth or soon after birth in the human body, but they are not excellent carriers of oxygen. So that is why most of the time the prenatal or neonatal organism depends most of the time hydrogen which is present in the maternal bloodstream. So most of the oxygen is coming from the maternal bloodstream. Hemoglobin in fact show a positive cooperation. We will talk about this positive cooperation in a short bit of time. Most of the time hemoglobin, so this is the structure of hemoglobin and myoglobin. Both are in fact oxygen carrying molecule. We know that hemoglobin has 8 helix structure, and in between them, and 8 could bound to this porphyrin ring I have also shown you, and this could also carry an oxygen molecule, and it could most of the time myoglobin carry oxygen to the muscles. The bad thing is with myoglobin, it do not release oxygen quite easily. So that means it could carry oxygen to the body parts, but it do not release them quite easily until unless there is really a stringent condition or really an oxygen depleted condition, then it could release the oxygen as is the case in muscles. So these are the 3-dimensional structure of myoglobin and hemoglobin. There is although their structure is quite similar to each other, of course the hemoglobin molecule evolutionary is conserved in all organisms. When you talk about reptiles, avians, and mammals, but still there are some invariant residues. For example, 9 residues has been shown invariant in hemoglobin, and there are other residues which are conserved. So several interactions are present in the when you talk about the different oligomers of the protein. For example, oligomer alpha are having various non-covalent sites. Several of the invariant amino acid residues are present somehow in this pocket where the heme molecule could bound. That means these 9 residues are invariant residues. They are very important for oxygen binding, so that is why they are still there. Other residues are there to stabilize the alpha helical structure, and there are still other molecules which are important and to develop this hydrophobic core structure. If you still remember, जो हमने पिछले lectures में बात की थी कि जो globular protein है, most of the globular protein, they are arranged in such that hydrophobic residues have joined together to make a cage-like structure, and because of this cage-like and this is most of the time buried inside the protein structure, and this make it possible for various active sites to play their role. And so this is that is why these chemical reaction, these quaternary structures are very important, because they make proteins functional. This is how hemoglobin looks like when it is bound to oxygen and when it is not bound. This is a gift structure which tells you which is in between these two structures. Down there are two different structures shown here. One is oxyhemoglobin structure, another one is the deoxyhemoglobin structure, as is shown here. By the way, by nomenclature, we say oxyhemoglobin and R state, that means a relaxed state hemoglobin, and the one which is without oxygen, this is called the taut structure, that is a tense structure that do not bound with oxygen. In both of these structures, you could see the beta change up and alpha change down there. And this is very important to understand that as soon as the oxygen molecule bound to this hemoglobin, one of the heme molecule, the structure of the alpha and beta is slightly changed. You could see, I mean, of course, there is a major difference, but you could focus on this. For example, my cursor is there on this place. You could see the loops and helix has somehow changed their structure. Similarly, you could go on the bottom side of the slide. You could see these two areas. They are slightly different from each other, and of course there are other changes which are, so that means there are slight interaction change. As soon as a ligand could bound to the protein, there is an allostatic changes. That means this protein is getting changed, and as it is obvious from this how is called this animated structure, where once the oxygen is bound, it changes its shape. This is called allostatic structure. Uh, by convention, as a reality, uh, as soon as an oxygen molecule bound to hydrogen, the relative position of two alpha and two beta chains change slightly. This structure shows this picture shows that as soon as oxygen molecule bound to the hemoglobin, and beta subunits could slide up to an extent of 15 degree. So that means when previously it was like this, and now they have somehow changed their position. So this is the change of beta chain which slided like this. So this change is very important because as the first oxygen molecule bound to the heme present in the hemoglobin, rest of the three positions are exposed for further binding. I mean, there are four positions for ligand binding in hemoglobin. As soon as the first bind to the oxygen, the rest of the three could easily bound to the oxygen because this is a positive cooperativity. Binding of one oxygen just slide in a way that it makes it possible for the other heme molecules to bind to the oxygen. That is why it is a positive cooperativity. So this is a conformational change that make it possible for further oxygen binding to the hemoglobin molecule. In upcoming video, we are going to discuss allostatic mechanisms of hemoglobin molecule and how enzyme kinetics are affecting these transitions. Moreover, we are also going to see how Hill equation is more suitable to explain sigmoidal oxygen curve than normal Michaelis-Menten equation. For further videos, please keep on watching.